Thank you very much, and I've got to say, it's really inspiring hearing all of these talks, and I hope I can do this one justice. And we're taking a little bit of a different tack here. I'm talking about a classical diagnostic tool doing DNA molecular diagnostics, and then looking at ways in which I can then spread them throughout the world and connect them. But the main point of this is that we are doing DNA molecular diagnostics to a clinical standard, to a referral laboratory standard as well. So a little bit about my story and the company's story. I am a molecular geneticist. I studied Drosophila fruit fly and was very passionate about genetics. This is because I'm colorblind, I'm dyslexic, so I was certainly incentivized to learn a little bit more about the genetic code. It's a lot easier for me because there's only 4 letters, which is really, really helpful. One of the problems, however, was studying. They wouldn't let me drink beer in the lab at Harvard, so I had to set up my own laboratory in my garage to do the kind of research that I really wanted to do. Again, the problem with this is I couldn't get all the chemicals and the equipment I needed. Number one, because I couldn't afford them, I was a student. And number two, you generally don't get the chemicals sent to a home laboratory. So I spent some time designing my own equipment, building the blocks of the molecular diagnostic process, and really playing and having fun, studying the Drosophila fruit fly that I was growing within my garage as well, and trying to understand the genetic variability within them, just for my own kicks and fun, really. But then something happened. I flew to a conference in South Africa, and when I was there, we were taken out on a township tour, and I saw the effects of infectious diseases that were whipping through communities, decimating families, and really causing serious issues for them. And there was no real diagnostics there. What we could see was that we were taking samples, sending them to the laboratories, or using lateral flow at the point of care, and it really wasn't having that much of an effect. So that was a light bulb moment in my mind. I knew that with the technologies that I had been building in my garage, the low-cost technologies that I could access just for my own fun, I could then put together and possibly create the kinds of diagnostics that these people need. So this will be DNA diagnostics for infectious disease that can look not just at whether or not you've got the disease or not, but also looking at drug resistance as well. And of course, my main focus initially was in tuberculosis when I was in Cape Town. Now, I didn't leave Cape Town for 2 years. So I stayed there, got a lab in Stellenbosch University, and started further developing this technology. And I got it to a point where I got proof of principle, and that attracted funding from the UK government, from the Framework 7 grants, and from some high network individuals. And I was able to build a company. And we've been building this company for the past 8 years now. And so we've got offices and laboratories in Singapore, Cape Town, in America, and in Newcastle in the north of England. We also have partners all over the world. And so we're now addressing the significant problems of infectious disease. And I don't want to think small. I don't want to deal just with the patient-to-patient, patient-to-doctor, relationship. I want to think globally. I want to think public health. I want to think global health. And so what we're trying to do is create a network of devices so that we can monitor not just what that patient has, but also what is happening in the world. And I'll get onto that more in a little bit. But the problem with the diagnostic process at the moment, especially for molecular diagnostics, is that it just takes too long. Now, when we're thinking about doing diagnostics in Africa, for instance, some people have to travel days to get to the clinic, and then you'll never see them again. So you need to get an answer then and there. Also, they cost too much, and we need to bring the price down. So another thing you've got to think about is emergent diagnostics. So what's wrong with this picture here? It says day 112 into the swine flu epidemic. And we're only now picking up confirmed cases. And as you can see, that's too late because the infection spreads throughout the world rapidly after that. And the issue is you have to know what you're looking for. So one of the problems with the diagnostics, molecular diagnostics for flu, for instance, is you have to know the sequences, and you're actually amplifying for a specific known disease, not for unknown. So again, I want to address that. I want to be able to pick up the unknowns. For instance, emerging epidemics that we've just seen in West Africa, the Ebola. But then also, it's not just that. We've got issues surrounding the drug resistance. So here you can see this map as it develops through the years. You can see that suddenly Russia started sharing data. Suddenly we started to understand that there's not just a problem with the drug resistance, but then also with how it's managed in terms of being reported. And so the problem was much bigger than we first thought. And before we knew it, we had issues. And this has a significant impact. When you think about the swine flu epidemic, it costs a lot of money. Furthermore, when you look at the drug resistance, the treatment of drug-resistant tuberculosis, it increases significantly when you go to multi-drug resistance and extensively drug-resistant TB. And this is more, there's a recent review just being released in the UK that has estimated if we do nothing about antimicrobial resistance, in 2050, it will cost the world $1 trillion. And it would also kill 10 million people every year. And that's a significant problem. So what we want to do is we want to shorten this time. We want to deal with these infections when we're in front of the patient. And then we want to share the data as well. We all know the internet. We all know the Internet of Things. I learned about the Internet of You today as well. And what we're trying to do is link the genetic code with the binary code, with our device in the middle acting as a bio API, translating this genetic code to the binary code. And if we have enough of these devices around the world, sampling the populations at different time points, sequencing the DNA, geostamping it, sending it to the cloud or to heaven, then we have an ability to monitor what's going on. So we have an early warning system for emerging pathogens. We also have a research tool where we're gathering data, sending it to the cloud, so we can understand drug resistance in a way we've never understood it before, understand the dynamics through populations. And we can also get resources to those areas. Furthermore, we can start managing our anti-infectives better. If we know what a bug is resistant to, we can give the right treatment, and therefore we can manage what we already have right now. To develop more drugs, it's going to take a long time, 15 years. What happens within those 15 years? Well, we have to manage what we've got, and using a sentinel diagnostic like this tool will help do that. So, moving on to some technology, and this is where it gets interesting for me. So think about the molecular diagnostic process. You've got, for instance, a whole blood sample. You need to get rid of everything else than just the DNA. You have to then make lots of copies of the DNA, specifically mutations. You then have to find some way of detecting it, and in some cases, you need to sequence it as well. So we've spent some time thinking about this. We took a step back, and we thought, how can we do this quickly? Generally, to sequence from a whole sample will take a week, for instance, including travel time, processing time. You'll need lots of infrastructure. You need a skilled workforce. You also need lots of expensive equipment and reagents. So we've had to take a step back and think, how can we do this cheaply? If we're selling to the developing world, we need to make sure that we have very low-cost devices and very low-cost tests. So everything that we've been doing over the past 8 years is making sure that we develop technologies that are off-patent or are our own, so we don't have any royalty stacking. One of the first things we did was create DNA extraction technology. So unlike the standard paradigm of DNA extraction, we actually keep the DNA in solution. So once we've done a lysis step, where we have a soup of different chemicals, proteins, lipids, etc., and the DNA, we push it through our filter, and it binds everything but the DNA, and the DNA flows right through. And this is revolutionary because it takes 2 minutes from its sample input. As you can see, it actually provides us with more DNA than a standard QIAGEN spin prep, which is the gold standard for DNA extraction. And this is important because we have to ensure that we get very low sensitivities. And so if we're not losing any DNA in the DNA extraction device, we have more chance of actually picking up the low copy number bugs. Then PCR. We all know what PCR is. It's the amplification of specific areas of DNA. This generally takes about 30 minutes to an hour, but we're able to do 35 cycles in 4 minutes. And this is by, rather than heating and cooling a heat block that heats and cools the PCR mix, we simply take the PCR mix through different temperature zones. This creates a thermal cycling, but at speed. Polymerases usually work by incorporating 1,000 nucleotides per second in nature. So we've taken advantage of this efficiency and this speed by creating the thermal cycling, which will facilitate rapid cycling. Then when it comes to detection, we are able to amplify many, many different amplicons. We can multiplex our PCR significantly. So therefore, we need to have a methodology of detecting lots of different sequences. So we've decided to borrow a technology from the computer field, a silicon chip. So we're using field effect transistors, silicon nanowire field effect transistors. These nanowires are 35 nanometers in diameter. To put that into context, you can fit 1 million of them to a human hair. When we have our amplicons, we flow it over the nanowire chips. And on the top of the nanowires, we have probes. When DNA hybridizes to the probes, as it's naturally negatively charged, it changes the charge density on the surface of the nanowire, which provides us with a signal. And that tells us that the nanowire is on. So we're converting the DNA into binary code directly without any other markers, 1 to 0. We can take this further. We've created nucleotides where we are able to flow them in one after another. And these nucleotides have a very high charge mass associated with them, which again turns on the nanowires as they're incorporated, which allows us to do sequencing as well. So we can amplify many different amplicons, and then we can sequence the amplicons as well, providing massive multiplexing capabilities. I won't bore you with all of the technical details of this, but essentially this is a raw data script from one of our nanowires. As you can see, it's very noisy. So my company, we have physicists, we have biologists, we have chemists, we have data miners, and we have engineers. And we need all of these people to come together to work as one to create this device. And we've started building these devices. These are some of the early prototypes of our cassettes. So this is real, it happens, it works. And we've started building the devices around it. And we can see, as we're going through different iterations of our devices and the development process, this takes time. It's taken us 8 years to get to this stage. But I'm happy to say that we are. Those eagle-eyed among you will see that sat on the bench here is one of our devices. This can sequence DNA from a whole blood sample in 15 minutes. We're now starting to look at trials. We're setting up trial sites in Gabon, in Southeast Asia, and elsewhere. And this is not going to just test whether or not our devices work within these environments, but we'll also test our idea of the Internet of Life, where we'll be taking this data and we're sending it to the cloud and observing how the public health system uses this data, how epidemiologists use this data, and whether or not we create any significant good from it, and how we can do it better. Obviously, this needs cooperation. So we're working with a lot of different people, from governments to NGOs. The Clinton Foundation have been wonderful to us, as has the Gates Foundation, etc. Without that, we wouldn't be able to do what we're doing. And part of that is to engage with people, to speak to thought leaders like yourselves, to get you part of this project, to help us out, because we can't do this alone. We've created an amazing tool but we need to create an ecosystem around it as well. So I spend a lot of my time traveling around the world. You may have guessed I'm a little tired at the moment. This is my 5th talk this week. And I talk to people. I'm trying to get people to get enthused by what we're doing. We have the chance of saving 10 million people a year with this device. But we need your help. We also need to think outside of the box. So I've been visiting some very smart people throughout the last couple of years to try and help me understand how I deal with this data. I'm a geneticist. I don't know anything about big data. So I'm talking to lots and lots of people. And of course, we've got to change policy, not just in Europe, but around the world. So we've been doing a lot of talking to some very smart people in government, trying to help drive their knowledge and help them see what the future is going to be like. And I'm now sitting on some government and international panels that have been set up to combat antimicrobial resistance. And these are really, really important because this puts our issues right at the forefront of government policy and international policy. And we're actually hoping that this issue will be picked up alongside global warming in the G8 summit. So that's what we're doing. That's been my past 8 years putting this project together. We've succeeded in making the device, which is one challenge. The next challenge is to get the Internet of Life set up, to get these out through clinical trials, and to start saving people's lives. And then we can see a future where we have these devices and other devices like this. It's not just going to be ours. But they'll be spread throughout the world. And we'll be constantly monitoring both the emergence of new infections and the emergence of drug resistance mutations. And we'll be able to understand the dynamics of these mutations and provide a sentinel for tracking and then curing the issues of pandemics and drug resistance. Thank you very much.